So good evening, everyone, and welcome to the 12th year April edition of the Sprouts lecture series. We started this lecture series with the purpose to, get people to become aware about conservation issues, about species and biodiversity, about landscapes, also to share ideas about livelihoods, the different kinds of occupations that are associated in the conservation field. When we started the lecture series was in person. We used to hold it at the Maharashtra Nature Park in Mumbai, and COVID pushed us all into our homes, and then the only alternative to continue this lecture series was to go online, and that of course enabled us to have speakers like we have today, Dr. Vishal Bhave, who is now sitting in Ratnagiri, while all of us are in different parts of the world. So that allowed us to get international speakers, speakers from across the country, and subject experts, who to bring them to Mumbai would have been very, very cost, uh, rather not cost effective for us, intensive in terms of cost, and logistically also it becomes difficult. So this has given us the advantage of having speakers from various places and participants also joining in from various parts of the world. Uh, the other thing is that this lecture series has not only focused on science, the scientific approach of conservation, but we also looked at parallel approaches, where we looked at literature, popular literature, children's literature. We also looked at some parts of mythology, where Devdutt Patnaik talked about how nature and mythology are kind of interlinked from one of his books. We are trying to explore even other forms, hopefully someday, where music and dance and theater also can be incorporated in our lecture series. But yeah, largely we stick with species science and of course community conservation as well as legal issues that are there. So we've had a whole diversity of speakers over the last 12 years. It is difficult to keep this session going both without good speakers and without good listeners. So we are grateful to all those who've joined us, and we have had a large bunch of registration, and we are hoping that they will join us very soon. So now I'd like to invite my colleague and friend Rahul Palekar to formally introduce Vishal. But thank you, Vishal, for accepting. We've been going back and forth for a long time on this lecture thing.
Yeah.
So I'm glad that the minute you landed back from Lakshadweep, you gave me a positive feedback, and here we are.
Thank you.
Yeah. So
What?
Rahul, please introduce the speaker.
Um, Thank you so much. Good evening, everyone. It is my honor to introduce Dr. Vishal Bhave to you. Dr. Vishal Bhave, Assistant Director, Marine Conservation at Srushti Conservation Foundation, is a dedicated conservationist with doctorate and postgraduate degrees in zoology, oceanography from the University of Mumbai. Since 2009, he has contributed significantly to conservation and research, focusing on habitat and species inventories, rapid biodiversity studies, and habitat restoration, especially in mangroves and corals. Vishal's expertise lies in marine invertebrate taxonomy, particularly in mollusca, and he is skilled in GIS and remote sensing for conservation applications. His work has been recognized with awards such as Sanctuary RBS Young Naturalist Award in 2009 and the Species and Habitat Warriors Award in 2023. With a passion for using technology to aid conservation efforts, Dr. Bhave's contributions to marine conservation and research are invaluable. With this, I would like to invite Dr. Bhave, and stage is yours.
Sorry, Vishal, before we start, I had just had a few housekeeping announcements. To all the participants, please keep yourself on mute. If you want, you all can keep your video on, but if you keep it off, it'll distract the speaker a little less. Secondly, Vishal, just one quick question with you. Would you want the participants to ask you questions while you're speaking, or would you prefer it at the end of your session? How would you want it?
In between also, it's
Right.
okay.
Okay. So I request all the participants to please type in your questions in the chat. I will be moderating it and maybe sometimes combining a question or two together and presenting it to Dr. Vishal here. And in case I feel that some things need a little bit of elaboration, please excuse me if I raise my hand in between and stop you and ask you to describe certain things more, because sea slugs, of course, very beautiful, but I think there is a lot to learn about the taxa, and yeah, we're very excited for this talk. So over to you.
So can I share my screen?
Yes, please go ahead.
Okay. Mhm. Good evening, everyone. I will speak on Sea Slugs of India. I will just introduce wh- what are sea slugs. So sea slugs are gastropod, just like whatever we see on our seashores. Those belong to same group, but as you see here, they have various colors and various forms, and the shape is also very much different than typical snails, means with shells. So those are classified in heterobranchia based on recent DNA-based studies, and this particular group has lost their shell during the course of evolution gradually. Initially, shell is thin and translucent, and uh, during uh, okay,
Yeah, please go ahead.
and in newly developed species, it is actually totally lost. So in this particular group, there are no shell is present except in their larval forms. So most of all the species have their shell in larval stages, but it is not present in adult stages. And in worldwide, they are present in large numbers in shallow areas, and in all there are about 6,000 and plus species, valid species of sea slugs known. Only two or three are found in fresh water, others are in marine waters. So in case of India, there are only 618 species recorded till date, and recently there is new species described by ZSI scientists, which is named after our uh, President Draupadi Murmuji. So it is this species, and only 150 or less species out of 618 are recorded more than 5 times. So you can understand, rest of the 300 and odd species are recorded single or less than 5 times. So there is no consistency in the records. So there is ample amount of opportunity to study this group. Most of the records belongs to Gujarat, Tamil Nadu, Lakshadweep, and Andaman Nicobar group. And oldest record are from Visakhapatnam and Chennai. So all this area, Tamil Nadu, Andaman, they have coral reef common in them. So all coral reef areas are already rich in their biodiversity. So rest of the areas have now being studied by many scientists and naturalists all along the coast, uh, and even in citizen science. So there are many species coming up from these areas, which were conventionally less studied. So what makes them so special? Their shape, their defensive strategies developed, their adaptations due to their losing shell, and their food specificity is very much important. So as I have explained this, their shell is already reduced, and in this forms, the shell is absent totally. In this particular species, shell is internalized as a thin, paper thin shell is present inside this body. If you touch this slug, then you can understand shell. else it will look as a blob of jelly. The feeding apparatus, which is known as a radula, which is comparable to our teeth, and it also is adapted based on their feeding preference. So this particular slender form, known as aeolid, feeds on Nidarian host, and as it feeds on Nidaria, most of the hydroids and soft corals and other group are thin, slender in shape. This particular species developed in a slender shape. So this particular species feeds on these polyps of hydroids, and this is jaw, which is equivalent to our mouth. It cuts. It is means uh, scissor-like sharp edges are there to jaw, and two jaws cut this polyp, and this radula, which is single line in a single line, it scrapes off whatever the tissue material from that polyp, and it sucks it into mouth and other further into digestive system. In this particular group, which is known as Sacoglossa, which feeds on algal material, various type of algae, not any specific, but Caulerpa or related groups. This is Elysia, and it feeds on by puncturing cell wall of this uh, algae, whatever the host algae is, and it sucks out in all the green material. So it has developed its green colored, uh, and it stores all chloroplast inside its body. So it uh, feeds on green algae. Based on color, you can identify where it may have its food source also, depending on whatever is inside its body. This third type is doride form, which feeds on sponges. And as you know, sponges are sometimes spread thin on uh, underside of boulders, or sometimes there are barrel-like formations on in reef areas. So they have can't use such thin radula to scrape off these surfaces. So they have developed this array of teeth along inside their mouth, so they can scrape off much more surface area of sponges and grab as much material as they can during the feeding phase.
Vishal,
So
Ushaji is asking whether that was the feather algae on the left bottom in the earlier slide.
one how to go back.
Can you go back?
Which slide?
No, the earlier one was there a feather algae slide before this.
Which, uh, wait, is it visible? This
Yeah,
one.
this one. Yeah,
This one is Caulerpa
oh,
scalpeliformis.
right. Okay, sorry. Go ahead, please.
Okay. So video playing. Video, some error, technical error. Okay. So if you concentrate on this particular polyp, this species will feed on this polyp very soon. So you can understand how it feeds. This video is quite old, so quality is not that great. I I can share the link of this video to Anand sir, and you can spread across. So if you see this particular strand, there was polyp, now it is almost gone. So all these species coexist on this two hydroid. That is also due to the evolution, and they prefer these two particular uh, hydroids as their food. So we can find many species on single hydroid also.
All other participants are requested to please keep their things on mute. Thank you. Yeah, Vishal, go ahead, please.
So these two species of hydroids, Penaria disticha and this is particular Eudendrium, both have are distributed all across coastline of India, and most of these species like Cratena and this Fidiana both feeds on these hydroids, and those are also found all across the coastal area of India. So this particular species Sakuroides gujaratica and this Phaseolina lineata both only found in Gulf of Kutch area, uh, like Poshitra or Narara, these areas only. Other side, it is very rarely found. So hydroids may be distributed all along coast, but this particular species are not distributed. So some are means that why it is not distributed, it's still not known even after monitoring it for 5 to 6 years. But we are not able to crack why they are not existing, and means at least around Maharashtra coast, which is nearby Gujarat, or other coastal areas of other countries like in nearby Red Sea, because all other species in Gastropoda are distributed till Gujarat, and there is some break. So this is also one important question about their distribution. They have Sea slugs have developed defensive mechanism as they have lost their shell. So they have cryptic nature. So if you can see here, they have a similar pattern as it is on this host bryozoan. But you can identify this particular species based on presence of other marks like egg cases or feeding patterns on the bryozoan. So it is easy to find. So other species, this one is Elysia pusilla, which is exclusively found on this algae Halimeda, which is found in coral reefs. So it is also very small sea slug, and we cannot see it if we look algae from a distance. But if we look it closely, then only we can identify this particular species with the help of these various marks on the algae, which are feeding marks. So here, this particular species is camouflaging Ocenia pilosa. It is always found on this hydroid, not hydroid, bryozoa, which is under the rock, and there are 9 individuals which can be marked, which are marked here as a red. So there are many such species which are not easily distinguishable or uh, easily viewed, but they are there silently present on all along our coastline. So, and this is orange color sponge on this particular area has uh, one sea slug, Rostanga species, which is a dorade, that is sponge feeding sea slug. If you see a specific sponge from a distance, it is not readily visible, but if you see it from near, then you can view it. So it is a good example of camouflage behavior. This is defensive mechanism with secretion of various chemicals. They can secrete deterrent chemicals, and like this white cloudy substance. When we collected it, it created this white cloud-like structure, so that it, because from that animal's perspective, we were predator, and that's why this behavior is seen. This is also a similar kind of cloud. In this aplysia, which is sea hair, they create such clouds of inks, just like octopus and other squids and related animals. So they can they can't actively swim, but they use tidal currents or other water currents to get away from their predators. Another mechanism to protect the self is by harvesting stinging cells from the host cnidarians. So these cnidarian host have their own protection mechanism. This species, which feeds on that hydroids or corals or other cnidarian host, they harvest these stinging cells, which are known as nidocysts, inside their body and store it in this nido sac. And they keep it uh, in such a fashion that they are stored as an active. This, those are not in a fired state like this. So this is how those nidocysts look like diagrammatically. So once they feel that there is some uh, predator or some danger, they release this neurocyst into nearby water, and the predator or that danger can get attacked by this neurocyst, like barb-like things, just like stings. Autotomy, like in geckos on land, this is similar to that. That tail is amputated during predation, or like similar to that. This particular group of species can shade their serratal bodies, and those are kept alive for at least 3 to 4 minutes, so that that predator can attack those particular bodies, and the other rest of the animal can stay safe. Breeding congregations are common in many of these species, and this Stylochilus, then Dolabella, most of the sea hares are commonly seen in large numbers in the habitat during certain days of the month. So they have breeding congregation, they breed, they lay eggs, and they vanishes off. So during peak period of means whenever the food availability is more, they do this kind of behavior. Other thing is solar powered sea slug. This is popularly known species Elysia chlorotica, but other species are also uh, similarly this Elysia, other Elysia and Costaciella, and that algal feeding group can uh, yield active chloroplast into their bodies and keep it alive for some days to manage production of glucose. They may not be self-sufficient because they actively feed on algae also, but partially their need of energy is managed through chloroplast. So, so whatever variation we saw in this green colored sea slugs, it is mostly because of feeding behavior. Some of the starving individual may look paler, and actively feeding individual may look more bright green. Uh,
It's a nice break. Go outside and walk and come.
They have developed defensive mechanism in the form of actively storing spicules from sponges and other bryozoan. It is especially found in some of the dorids, and they can, if you can make out here, that's silvery or this straight rod-like things, which will look under microscope like this. They create endoskeleton using those spicules. Those spicules also help because whoever feeding on them will not like some spiny things inside their mouth, so they deter them from predation. These are rhinophores of various types in various group of animals. Rhinophores are basically sensory organ developed specifically uh, to detect uh, mate or food or other animals, because their eyes are, as you can see here and here, those are just spot which sense lights. So they have to develop their own uh, sensory system. So these are used as a sensory organ to uh, find food and mate and other things. Gills are also specifically developed and specifically aligned. So fan-like, it can be vertical rosette form, or it can be laid on only on one side, as the case in most of the side gid slugs, which are belonging to Pleurobranchomorpha group. So, and there are many type of these gills, means with single branch or with multiple branches of small gillets. So depending on that, we can these characters are basically taxonomically important, so we can identify particular species and the habit and a habitat. Depending on that, the gills are developed in such fashion. Foot and other structures. This is a dorid which found underside of boulder. So their surface area is more and it's more dorsoventrally fattened, and their foot is broader than eolid, which is found feeding on hydroids. So those their shape and structures are different. As earlier told, these are serrata and medosac. They can shed off the serrata. There are different type of serrata in eolids. Depending on that, we can find certainly find and bind them to specific genera. They have different reproductive strategies also, but I am not going into that in detail. But from our perspective as a citizen, we can find these various egg cases and types of egg cases, and based on that, we can park them into certain genera or group of individual particular species. So where we can find this? So what are their main habitats? So
Vishal, can you just go back on that egg slide? Sorry. And
okay,
why, like the different structures that are there in the laying of eggs? Is it related to body type or any other?
yeah, this uh, most of them are in rosette shape. Even in means groups are different. That nudibranchs, dorids, it is in specific rose-like form, and their shape is thick, and most of these structures are underside of the boulders. Those are very well protected. This Polyerpa and Sacoglossans also lay their egg on specific means they are host animals. This is hydroid, and this Doto species lays egg on the tip of hydroids. So depending on where they lay eggs, means on that how it is formed is there. Then this Maggie-like thing, noodle-like, is from anapsids, means sea hares. whereas sea hares are bulky, as you know, that ink throwing animal. So it is quite large, so it lays egg in such fashion.
Excellent.
So most of the species are found in coral reef and rocky patches. Very few, means 5 to 8 species, what I know, are from mudflats in India. Most of species recorded from Maharashtra are from rocky shores. Very few are recorded from mangroves. Sandy shores, we don't have, from my understanding, any species yet. But there are many species which live inside sand. So those are not found during daytime or during low tide. So we have to actively search them during high tide and all. So those are a little bit difficult to find. So uh, we lack records of this particular habitat from our area. Sea grasses, there are few species which feed actively on sea grasses, and those are found on particular species of sea grasses. Each office sovereign, as I told earlier, has specific food requirement, and thus their habitat preference also differs. So most of the sponge feeding sea slugs are found underside boulder because there is ample amount of food underside it. So most of the sponges are found underside boulder. So uh, availability of food material is there. So dorids and other similar kind of sea slug which feeds on bryozoan, sponges, and whatever grows underside, they are found in that particular group habitats. So this is underside of one of the that boulder, and there are hydroids and sponges which are found easily. So there are this hydroid loving species also found near this under these boulders. In small standing puddles, we can, in mangroves especially, we can see this Elysia. Two species are recorded from India. One more species is recorded from India, from Andaman Nicobar, which is very small, but it's very hard to record. These are one of the common species of Elysia. Many species which feeds on hydroid and bryozoan live their entire life on that. They lay egg on that particular hydroids, and they cover almost all their adult life on same area or same pond or same hydroid. So if you see this particular species, which is find in between this gaps of zooantherians, which are again nidarians, they also harvest some xanthily from that zoantherian host, and they have used these areas only for laying eggs and feeding and doing all their active life, except their larval life. So next, all the next slides are just photographs. I haven't named them all because it will be yes, become more complicated by names. That's why. So these are basically bubble shell slugs. This is very small, less than 3 millimeter. If you see morphologically, bubble shells means cephalaspedians are head-shelled slugs, as scientifically known. They have parapodial lobes, external shell, head shield, and eye spots. Some of the species have shell, internal shells also, like this particular species has an internal shell. They have foot and they have different colors. Means sometimes colors are brown, black, and dark. Sometimes white, and most are means vividly colors. Those are specifically found nearby sedimented rocky shores, and sometimes on algae. This acetosina uh, is found only in sandy patches and with algae. This group is found on sand, which lacks shell, or shell is sometimes inside their body, not externally visible. These are sap-sucking slugs. Sap-sucking slugs are known as Sacoglossa, which most of them are feeding on green algae or other type of algae. And morphologically, these are these have parapodia, which is large, where they store their uh, whatever material they feed on, with along with chloroplast, and rest of the area they have head and other systems. Some has a external shell, and others they don't have any external shell.
So Vishal, there's a question asked over here by
Okay,
Usha, which is that how did sea slugs lose their shell? What was the cause? Do we know anything about it?
cause it's not yet known, means
Okay.
easily, means I cannot tell you, like, means I don't have any
Evolutionary
answer to
studies
specific
need to be
question,
done.
because
Okay.
based on the evolutionary tree, it is told in various papers, but it's still work in progress, I
Right.
will say.
Yeah.
So,
Would you take two more questions at this point, or you want to do it later?
no, I can take
Okay.
questions.
So one is a repetition from what you already told, but they were asking that where are the most number of species found in India? In which place or
In
which?
coral reefs,
Okay, and which part of India?
means out of India, means
No, which region, like Lakshadweep, Andamans, or where?
Lakshadweep, Andaman, all most of the reefs area has large species.
Okay,
So, and colorful species. So those are easily found by any diver, whoever goes in that particular habitat frequently.
okay,
Other areas has means that cryptic and other species are very difficult to search. So those are not easily seen and recorded.
okay. Yeah, please go ahead.
So these are sap-sucking slugs, Elysia group. There are quite a few species recorded from India. There are many more species yet to be recorded. These are just few examples
If you
of
can go a little
group
slower, please,
Elysia.
on those.
Okay,
So,
okay. I
because,
will go back.
yeah, sometimes with network they tend to slow for people. Yeah,
Okay.
thank you.
This particular group is Elysia, which feeds on green color algae. Some feeds on other red algae also, but most of the that color is based on this green pigment. This is colorful genus Turidilla, which is found mostly on coral reef areas. They feed on algae only, but those can be found under boulders also. These are aeolidae form. These are not true aeolid, means those which feeds on hydroids or nidarian prey. These are feeding on filamentous algae or other type of algae, but they have developed their body in the form of cerata, and they store all whatever they suck from that uh, algal cells into these. So cerata in this case helps to increase their surface area, by which they can actively feed on more number of food material whenever it is available. So all these species are very small except for this species, which and this species, which is more than 2 centimeter. Rest of species, this group is less than 1 centimeter. So those are there are very few records of this species. Uh, these are shelled sea slugs from Sacoglossa, sap-sucking slugs. This Lobiger, Oxinary, Berthellinia, then Acetobula, then Cylindrobula, all this, and Berthellinia. This shell may look like bivalve, but it is developed in such a way that from small larval gastropod shell, this shape is evolved. So, in true sense, it's not bivalve, but it's just a shape is similar to bivalve. It also actively feeds on green algae. So sea hares, Aplysia, that ink, purple color ink, that video was shown. This particular group has a parapodia head and all other parts. This species of sea hares are easily found, but those are also cryptic when they are uh, inside that large mats of brown algae. Those are hard to find, and some of these species which are easy to locate in the reef areas, but on the rocky shores they are very hard to search. uh, Stylochilus striatus, that this green color one species which is Petalifera, I guess. Not Petalifera, I forgot its name, but it actively feeds on seagrass. It's if you see this photo, this same species which is spread thin on that green color seagrass. Then side grid slug, Pleurobrancomorph, they have internalized their shell. Then, as the name suggests, they have a gill on their side, and all this species has bright color, and they can secrete defensive chemicals which is acidic in nature. So most of fishes and other predators means don't feed on them. These are arminids which belongs to nudibranchs, uh, which are brightly colored, and they feed on various soft corals. Some of this particular group is found in on Carijoa. This is found in coral reef all across India. Means it is found in Gujarat, then Gulf of Mannar also, then Lakshadweep and Andaman Nicobar. This particular group, which is Doto allied Dendrodotonidae, it has a specific type of trumpet-shaped rhinophore margin from which the rhinophore arises. So, and they have developed their shape to cling on various type of hydroids and other nidarians prey. So they have the foot is thin, elongated body, and tail rhinophores and serrata. So, so these are some representative of this group, Dendrodotonidae. This particular species has a large mouth, which is in, they open it like net, and they feed. They are basically found on sandy area with sedimented area. So they means capture food from water column. Dorids, which don't have uh, retractile gills. So those are known as phanerobranchs, and their gills are over their body. These gills cannot go inside their body. So these are some representative from reef areas. These are non-reef forms which are found on the rocky shores. All these species which belong to Goniodoridae family, and and one is from Onchidoridae. All those are less than 1 centimeter in size, so those are very hard to find. And most of these species feeds on bryozoa or ascidians. These are gymnodorids. Gymnodorids are known to eat the fellow partner or any other sea slug which are kept along with this. So uh, those are also found in uh, various reef habitat specifically, but some two or three species are recorded from non-reef areas also from India. This particular group of dorids has gills which can go inside their body. They have uh, opening from which gills can come out in a rosette form. So like this, the gills can be taken inside. In this particular photo, you can't get see any gill because it is retracted. Here also, gill is retracted. All these are dorsoventrally flattened and has always feeds on various type of sponges. These are just representative, I mean, photographs. Uh, there are many more such species. This particular group of dorid with retractile gill, which are known as dendrodorid, belonging to family Dendrodoridae, has chemical deterrent compounds as their active shield, and they feed on sponges not by using radula because they don't have any radula or teeth. They are I mean, suctorial mouth, and they suck everything. And using their chemical compounds, they prepare slurry out of that sponges or whatever they feed, and they feed like that. So, Bromodorids. This is one of the largest family, means highly diverse family of sea slug, mostly found in reef areas. So they are beautifully colored, and they also has they feed on various sponges and other benthic, means interesting fauna. So some of the species are found in rocky shores, like this particular, then Bombayana, Bombay sea slug. Then these are from rocky shores of Dwarka, and there are other groups which like Hoplodoris, then Halaxa, then
We shall just go a
Joruna.
little slow between the slides because yeah, thanks.
Okay, so these are dorids. Other than whatever said earlier, so all other genera are I have segregated into this. These are Hoplodoris and Joruna, then Halaxa. All these are found on the rocky shores as well as these are reef forms. So they also feeds on various sponges and sometimes acidians also. These phyllidids are exclusively found along the reef areas. These are known to release deterrent chemical like that the ascobula white color, that mucus like thing. They also release mucus with very bad taste for predators. So they actually defend themselves using that strategy, and sometimes their color matches with some of the sea cucumbers which are found in uh, reef areas. So from that perspective, also some of the species that use that to defend themselves. all this phyllid don't have radula. They also use their suctorial mouth to eat their food sponges. These are aeolids, like told earlier. Those are nidarian feeders, and they actively feed on zoanthids of corals and hydroids. Uh, all these are representative of this aeolids. This particular species was described from India earlier, back in 1968, from southern India, by a scientist from CFRI known as Eubrancus mandavamensis. Some of these species are rarely recorded because only single or two individuals are found during the survey. So those are very small and not easy to dissect. So their records are not means published. So there are many such records exist in the repository, but they never published. This is also described from India and it's found all along Indo-Pacific, but it was described in 18, 1800s by Alder and Hancock, who described or studied our Indian sea slug fauna for the first time in the British era. So this is very much important sea slug. This particular species, which is beautifully colored, is found on a white colored carijoa, which is invasive. So this is easily found on that particular soft corals. This one is Sakura Eolish gujaratica, and this eolid is found exclusively in Gulf of Kutch, and it's endemic to certain areas of Gulf of Kutch. Though its food organisms, like are told earlier, that Tennaria and Eudendrium are found all along the coastline, but this particular species has not traveled along with that. The question is why, and it's not known yet. That's all. Thank you.
Thank you, Vishal. Please take a sip of water. We'll have a bunch of questions
No
that are there.
problem.
So we'll,
Okay.
yeah. So, there are comments like amazing pictures, feels like they have all the colors of the world. So yeah, indeed, it's a fantastic world. Uh, you had a sip of water. Shall we jump into the questions?
Yeah, we can.
Okay. So for first question, so what you talked about of the loss of shells, there's a comment on that about by I think 1 second, just give me a moment. Aditya said that did they lose their shells to move faster? Is that
Not necessarily,
not necessary?
because their speed is not fast as all gastropods don't move fast. Some of the species has the ability to actively swim. Rest of them other depend on currents if they want to move for a long distance.
You can stop sharing the screen so that
Okay.
then you can be in the center. Yeah, I'll just spotlight you. Okay. The next one is by Neeta Ganguly. She's asking, can you elaborate on kleptoplasty about sea slugs, in sea slugs?
Leptoplasty is that solar powered sea slug. So they harvest that chloroplast from
Chloroplasts.
that green pigment from that host algae, whichever it is. So they use those chloroplast. Only chloratica, Elysia chloratica is known to be actively photosynthesized. Rest of the species either are not studied in typical fashion or they don't actively photosynthesize. They can rely partially on that, but like Chlorotica, they can't.
So they're not exclusively photosynthetic.
No,
They
no.
also are heterotrophic and autotrophic at
Yeah,
the same time.
yeah. They feed on algae and they harvest whatever they can. They keep it. It stays alive in their body. Then, whenever it get destroyed, they refuel like that. Keep on going.
So one thing is very clear that I think if all the members of the audience, you should understand that there is a lot more to study in this taxa in sea slugs. That there is a lot you can do, and more you should more people should consider getting besides photography and just observations. I think people need to get deeper into the study of the evolutionary biology, the behavior, and the ecology. So I had a question, Vishal, that in terms of when we say an ecological niche, what would you say is the ecological niche of the sea slugs? Where do they fit in the food chain or in the ecological, pyramid?
It depends on different species because, as far as I know, they have very limited predators.
Okay.
I saw means last 12 years many times on field, but I only saw two times. One crab was feeding on Barsatella lychee when it was dead, not alive, means
Okay.
not killing it.
Mhm.
Whenever it was dead, like other that
Scavenging.
scavenging, yeah,
Okay.
like that. Actively, means someone is capturing and eating it like that. I never saw.
So what is the role that you think sea slugs play in the system then?
We don't know much of ecology of
Okay.
if I means
Yeah.
we tried to study one species, Sakura eulus gujaratica, for during BNHS time,
Mhm.
but uh, nothing was means came out of any study.
Okay. Right.
Yeah.
Satya Swarup has a very interesting question. Is that how do iolids and other species incorporate nidocytes into their cerata without triggering or digesting them?
They feed directly, like that video showed.
Ah,
They ingest entire thing,
okay.
then suck everything. They may have some chemicals to hold their trigger off. We don't know. Means there
Right.
are many research papers which are published, but I I haven't read exactly how that mechanism works. I was busy in identifying them, so
Identification. Yeah, somebody has to identify them at the end of the day as well, so that more studies can be done, definitely. Then Usha has a far more basic question. She asks that what are retractile gills? And then I would like to add and say, why do they need to retract their gills? Uh, what is, if you said that there are
so
not too many threads, then what could be the purpose for retracting their gills?
so that retractile gills species are dorsoventrally flattened.
Okay.
They are mostly living inside boulder fields, means large boulder underside that it can crawl. So that other species which don't have retractile gills, they feed on ascidians and uh, other groups, and their size is also small. This large versus small, maybe one means angle. I don't know a specific answer, but from my logic, I'm telling it.
Okay, right, great. Uh, Chinmay Sonamne is asking that regarding cryptic species within the genus Corambe, what methodologies do we employ for their study? Since I mistakenly identified them as flatworms. So I think, for people, common people also, how do you distinguish between a flatworm and a nudibranch? That would be, I think, question one. Then comes the second part of the criticism of Corambe. And if I can complete the question, may I? Or you want to answer this much and then?
No, you can complete.
Okay. I've come across your contributions of the Sea Slugs Forum, and recently I was fortunate to encounter them in Raigad. While these species are distributed globally, there appears to be a scarcity of studies concerning them in India. Could you provide some guidance or pointers for studying them effectively?
So first,
Yeah,
how to segregate between
segregate. Yeah,
flatworm and Corambe
beauty.
or whatever that genus.
Yeah,
So
nudibranchs, I would say in general, between
uh,
nudibranchs
eh,
and
eh,
flatworms.
so flatworms don't have gills or underside also. In Corambe genus, there is gill pouch underside near on the ventral side. So that is one. They don't have rhinophores or
Rhinophores.
rhinophoral cavity or similar structures, extrabranchial structures or anything similar to this flatworms. Flatworms have tentacle, but they don't have rhinophore-like structures with septa or other means patterns or constrictions or nothing like that. Only flat uh, structure.
Also, shells would be missing in any of their life stages, right?
Yes, but we don't know larval stage, right? So we get to know after they are settled and
Okay.
whenever we go on field. If we
But
go,
no, in the sense, flatworms wouldn't have shells at all at any stage of their life, right?
no, because these are gastropod and gastropod shell is typical with coil.
Okay. Spiral.
Yeah, so
Okay. Now going to how does one study in detail to do a detailed study on Corambe?
Corambe is, as far as I know, it is not found yet in India. But I am not means saying it. We won't get that because there are many species from Japan and other areas which have traveled to our shores. So it's quite possible to get that, but without seeing anything, I cannot comment on if it is Corambe or not.
Right.
Because that particular genre has that gills is typical underside, so and their shape is also typical, so you cannot means it will make out from other group.
So are there keys, Vishal, which are there, to come to, have you all reached the key stages till now?
Key currently that stability is not there to any molluscan taxonomy
Okay.
because of DNA and other newly developing means studies. So once database is fully developed and all the taxonomic tree is settled, at least at generic level, then we can, because every day names are changing. That's why I avoided all the names,
Right.
because if I tell some name and then it will shift to other name, next day it will be different. Who knows? Keeps on changing.
So also here at this point of time, you must be largely, focusing on the what you call, morphological features, because there will be no comparison for DNA studies also, right? There'll be no baseline for the DNA studies, or are there DNA studies being done?
DNA studies are means there are many species which are described based on DNA studies also.
Okay.
So integrative taxonomy is must first thing.
Okay.
If because without studying morphology, if DNA tree shows different structure, then based on that you describe species, and later we will combine that into some other genre because you haven't studied morphology.
But that's happening, Vishal, in many other
That's
taxa.
so
It is
it's of no
happening.
use.
Yeah, yeah. In
From
frogs and others,
my
people are
understanding
just
and
following DNA.
my philosophical view.
Okay,
I don't want to push it on any other person.
sure, sure.
That's
Okay.
why.
Usha has a very interesting question, and I don't know if that is, something that's been. But what is the effect of global warming on the entire group? And is there any studies started off on that?
In California, means American, that California coastline, there
Right.
are few studies which suggest distributional changes in sea slugs. I have a few papers on that. Uh, by, I guess, Godard and Ital. I don't know if I spelled means pronunciation is correct, but they have uh, used some modeling and distributional methods to deduce that some of the species which were present towards south now moving to north or north to south, like that,
In
because they
India,
have long term data.
in India, which were the oldest recorded sea? When was the first New Guinea
Alder,
described?
Alder, Alder Hancock, 1864. The Britishers,
Right.
they have described more
And which
than
area was that from?
2030. It, it is given as Whiter and Chennai. Their collection was made earlier.
Ah,
Then they used those material to dissect and do some studies. But all the material is available in that British Museum
okay.
till now.
Right.
So
Uh,
that is the oldest record what I can get back.
Come across.
There
Right.
may be other by means, which I may not have came across, but
But that
this
was
is
a scientific record or a natural history record?
no scientific record published in
Okay.
proper journal of that times. So
Right.
Annals of Natural History
History.
Museum, I
Yeah.
guess.
Yeah. Vimal from who's from Vizag is asking a very interesting question that if we spot a sea slug, what are the morphological and natural history observations one should make in order to identify and study it later? So if you could elaborate a little on that.
First, you capture as many photographs from all possible angles. Underside also essential. Rhinophores, front side, then back side. If gills are present, then gills whenever they are open. Then rhinophores, if they are erected, then because rhinophores are key character, gills are key character, number of gills, gill leaves, then presence or absence of extrabranchial processes, means whatever is growth or outgrowth of that uh, mantle area in the
Right.
form of tubular structures or serrata like structures. Those are also essential depending on means that groups which I showed segregated. Depending on that, structures are different, and those structures are essential to be studied. And another important note we need to make is where it was found.
The
What could be
microhabitat or
yeah, specific food, because most of the sea slugs are found on specific food material or nearby food material or wherever they lay eggs on.
okay. So another thing that I want to add to this is that should they sort of try to preserve them or should they try to keep them alive or they have to be extracted for studies like these?
If possible, and if someone is serious only, then they can collect. If just want for making a record or something as a citizen scientist, then they don't need to collect.
But
They can
permissions
take
will not be okay.
they can take photograph from multiple angles, which will lead to at least making a record of that particular observation.
So photos and videos is what we do.
Video is also because some
Some
species
behavior you might,
the behavior is also specific.
yeah.
I haven't added it. Some chromodorids shake their gills like this, vibrate.
Okay.
So that is typical to that particular. Some Hymenodias go some centimeter, millimeter forward, then come back again, then go again, just like
Okay.
other slugs
Peristaltic
on land.
movement. Okay.
Yeah.
Sure.
So like that, there are different behavioral
Any,
patterns.
see, typically in climate change discussions, what is happening is they're saying that sizes of butterflies or sizes of birds or sizes of mammals and all are changing. Do we have enough data to kind of make a directional thing that, oh, sea slugs are either growing bigger or smaller? Uh, because generally they should
Best
grow bigger if with, increasing carbon dioxide, the size should increase. So is there any, or
at
the data is too
present,
little?
I think their distribution will be changing.
Okay.
means from temperate area to tropical area or tropical area to moving towards temperate area, because temperature is increasing on near subtropical areas
Right.
due to climate change. So the other species which are food material of this particular species
Right.
will be growing there as per based on larval settlement. Because all most of the marine invertebrate larvae are planktonic in nature. So wherever they find good habitat, they will settle
Right.
with currents and all. So we don't have long-term data for any of this invertebrates. So
Right.
we can't comment directly from Indian scenario. But if we combine all global data and
Yeah.
plus our data, then and there is no means continuous data on sea slugs
Patchy.
all across
Yeah.
India.
Right.
So
So I want to continue on the question that Vimal had asked and ask you, have you published some protocol of observation as to what are the steps one should go through for field observation? Is there some document, any common article popular or something?
nothing,
And
nothing is published.
okay, so there is a potential to do something like that.
Yeah, there is much more potential. At least 150 species will be there, which can be easily found and described.
So that,
That much I know,
that
because
leads me to the next question which Usha has asked. Did you say there were 6,000 slug species or 600 sea slug species?
600 in India,
Yeah.
6,000 globally.
Globally. Okay. So
Of
which
which,
are the two freshwater species and what are their names, if you can, and where are they found?
if you go on Sea Slug Forum, you will get all the information,
Okay.
because
Okay.
I don't remember all means
Right.
freshwater species, because only two species are there, and those
But
were
which
not
area are
found.
they in? Any idea?
That I don't know, but uh,
But these are in Indian
uh,
waters or in
no, outside, not
outside?
in India.
Okay. There's no such record within India.
At least as of now, no record.
And in brackish areas, there are?
Brackish areas, that mangrove dwelling Elysia is
Yeah.
found, in other aeolid-like species of Arcolania, that is also found in mangrove areas. Three species of Elysia are there in mangrove areas.
Right. Amrita asked a question that since photography is an essential aspect in observing sea slugs, what are the ethical aspects one should keep in mind while clicking photos or videos?
Uh, Try not to remove it from habitat.
Okay.
If you are not serious scientist who is giving its life only on seas to do work on sea slugs,
Right.
uh, that's what I can say. But if you want to take photograph and keep it again back as it was, that particular animal from where you have picked, you keep it there so it can live its life naturally.
And Usha has another question where she asks that are there any guidebooks on sea slugs of India?
One book is there by Dr. Apte and Desai, I guess,
Digant.
Digant Desai.
Yeah.
So I use most of that reef photographs from them only. I don't have means that quality cameras to take photo
Right.
of, and I don't dive in the reef. So recently only I am going to Lakshadweep, so
Okay.
they have that
I
beautiful
think you'll
collection.
agree with me. I will like to add to Amrita's question and to your answer is that one of the ethical things is that if you've taken a photograph, then put it on a social medium like iNaturalist. Don't leave it in your system. It's of no value if your data is lying in your computers or in your phone and it is not there on a public forum for or available to researchers to be able to identify them or so I think that's probably the first ethical you owe it to science that you put it up for identification and share
Issue with that means iNaturalist, they have their protocol where it goes to research grade after number of clicks,
yeah
identify agree to
yeah
that particular genus or species.
yeah
Based on
that
that, it
system is
can
wrong but I'm saying at least the data is
no, you
available.
can put it, but if creates other issue.
Yeah, create correct.
So I'm trying to put whatever I can.
So
I already
what about
added.
something like Sea Slug Forum?
Sea Slug Forum is now dead
Oh, is it?
due to some means,
Okay,
whoever maintaining that Bill Rudman, who
yeah,
is I talked with him also
okay,
during that time,
right.
but it was not working. Now Nudie Base, I guess Nudie Base on
Okay,
Facebook,
right.
Gary is doing wonderful work. He's continuing that. Uh,
But I think this is the challenge of any databases. If people finally get tired or they get old, and then it has been handed
he
over
is
to
old,
somebody.
so no
Yeah,
issue is that every forum has to be answered by someone,
yeah,
and that his position is not taken by
purity.
other person, then it it is dead.
Yeah,
But it's now also it's one of the best resource available to learn about sea slugs.
right. I have a few personal questions which I'll come to a little later. You want to take a quick sip of water? There are one or two more
Uh,
questions. You're good?
no, no, no. I'm
Okay,
good.
great. Right. So Chinmay Sonavane has asked, do some species feed uh, animals other than hydroids and sponges? Like, do Anatolidalia species feed on sea anemone? Uh,
Yeah,
always found them close to some orange colored anemone.
all species have specific means their food preference is specific. It will feed on two, three species, but it can't feed on randomly on. If it's feeding on animal, it can't feed on directly to coral or something like that. So each species has food specificity.
Food
So
specificity.
Antivolidella feeds on various animals. That is correct. I showed only hydroid because hydroid are that
Easier.
evolid shape and easier to means visualize. Their slug is moving on hydroid and thin shape and everything.
Right.
So there are other species also which feeds on different kind of acidians also, bryozoans and other soft corals.
Also cannibalism, right? They also might feed on
Yeah,
their own
that
kind.
one genera that Gymnodoris, it's known for that. If you keep other species along with that, it feeds on
They
that
will.
also.
Okay.
So if we collect two specimens, only one will be there. So you
Hmm.
have to keep it on each one individual in single water.
Right.
Uh, Earlier we used to don't used to understand how it is happening, why our specimens are going
Missing.
missing
Right.
even after collection. So after learning on Sea Slug Forum, we came to understand that those are they have same behavior like cannibalism.
Right. Next question is again by Vimal. He's asking, are there any sea slugs which are protected under the law? Any scheduled species?
Nothing yet because we don't know anything about sea slugs.
But
Even
do
only
you think
recorded 600. I don't think it's needed for smaller species which are already cryptic, but and those are not popular in aquarium also because they are, due to their food specificity, those are not easy to means keep it in aquaria. Every
is there
time
a trade? Is there like like keeping ornamental thing? Are people keeping sea slugs in aquarium?
we can keep, but
Marine.
most of means on forum aquarium forum, those are treated as a pest
Okay.
because some feeds on coral. Then if there is coral tank, then the corals are dying.
Right.
So it is like pest to them.
Right. They consider them as best.
So, yeah,
Next question is again by Usha. What are you currently working on about sea slugs? Is there any trade, like what the question we asked, is there any trade of sea slugs that is happening?
as per my understanding, there is no trade, at least in India. But it's those are not easy to keep. That's why.
Ah,
Uh, and I'm not working on sea slug at present because it requires microscopes and other facilities, which my institution, we are currently building it. So we don't have it right away. So I'm not working actively, at least last few years. when I switched into SCS.
Then the next question is again by Vimal. Um yeah, I think the others also should ask questions. It's good that these guys are asking, but so do sea slugs feed on caulerpa, which practice kleptoplasty, as they are known to retain collar pin too? Or are they known to retain collar pin too?
Yeah, they written that particular means what chemical, and they also synthesize. There is whole
Okay.
one book on Springer on this subject, where they can means how sea slugs eat and produce de novo chemicals, which can be used as anti-cancer or other kind of. From our perspective, we are
Hmm.
using
Consumptive. Yeah.
can be used for some disease or something useful,
Right. Uh,
which
right. So next question is by Usha. Do they have any site specificity, especially for breeding?
most of them breed on means whatever they eat. So hydroid feeding slug breed on hydroids.
Oh.
So like that it occurs. Whenever food is available in large quantity, they breed, they disappear because their larvae are planktotrophic. So in plankton, they release eggs are released on that hydroid or whatever habitat. Then after that period, each species at different in 7 days, 10 days, 15 days, or whatever period. After that, larvae are released into water column. Then they settle based on next best season from their
Yes.
understanding.
Yeah. So uh, I have two questions here. One is that do sea slugs migrate? Is there any record of them migrating, and how, what distances, if at all? And of course, some most of them are too small. But can you fix any tracking device or something? Are there any of the bigger sea slugs? What is the biggest size of sea slug, and what is the smallest recorded?
So that see here, it's global size. I'm not telling
Ah,
for
yeah,
some Aplysia gigantea or something. If I
yeah.
remember it correctly, size is more than 3 feet or something.
Wow.
That's what
Right.
recorded
Okay.
means on the internet. I don't know
Ah,
specific, but as per internet that was given.
right.
But smallest is it lives in interstitial particles. So particles of sand. So it's like small worm-like animal, rhodope or some genus is there, which are brightly colored.
Uh-huh.
Some acocleidians are also in same, which are worm-like. They have radula
Hmm.
and some worm-like body. So based on that, those are classified because they have radula.
Right. So,
And larva shells is with having that spiral structure.
so the question on migration, do they migrate? Has records been? How would you track them?
Tracking is difficult because we don't have inventory first. So basic inventory is needed. That is, this is our indigenous fauna. Then only we can say these are coming from somewhere else, or
No, I'm saying
we
an
don't have.
individual sort of, this individual went from here to there, kind of. What is their home range or
It's
territory movement of an individual?
they that is not studied,
Not
at
sure.
least in India.
Okay. Yeah, I think the next question that Chinmay is asking is also interesting. Is that are there any deep sea nudibranchs? Like, what is the deepest point at which they have been located?
That is difficult, but I guess recent expedition in Philippines or somewhere they have recorded some species. I don't know exact means meters
Okay,
how much deep,
right.
but aphotic zone they have detected some species,
Okay.
but
In
those
the
are very
Philippines.
difficult.
Okay. Oh, wow. Uh, I had a more personal question as to how did you get interested firstly in the field of studying sea slugs and nudibranchs?
Uh, it was challenging. No one has studied. there was ample opportunity before coming of DNA into picture. So it was challenging. That's why
No, when is the first time you'd heard about it or seen one? How did you
in
stumble about
2009, I guess 2009.
it?
Yes,
Okay.
we went with Dr. Apte in one of the camp, marine camp in Malwan. As earlier also, I used to go and see some on rocky shore. Like now they are taking walks and all. I
Right.
used to go like mad person walking individually. My mother and father used to call me, "You are mad only." So like that, I used to went and whatever I find colorful, I used to bring back. I was not knowing that this is important, this is rare, or because internet was not flooded with information then.
Hmm.
Earlier, only one resource that Sea Slug forum was available.
Correct.
So I used to post it there. Then Will Rudman used to give very illustrative reply.
Okay.
That's how I learned. Then
So,
I meet Dr. Apte. Then I started my journey with BNHS. So like that, it went on.
so my second question is more personal again. Is that was it more fun and romantic to do this work when you were like a amateur and observing, or is it more fun doing it now as a scientist, as a researcher? Has the joy of doing that now it's very tedious and very pressurized, or how is it? Do you enjoy it more now that you are discovering new species or doing very, very detailed work? How do you balance?
Uh,
What has something changed, or
I
is it the same?
I'm not doing any work now. That is the
Okay.
issue. I had done means if you have all support needed, then you can do it.
Okay.
But at my position at present, I have to build institute again.
Okay.
So I don't have that luxury of time
Right.
because for dissection, you need full concentration, 2, 3 days at least. Because those slug size are 1 centimeter or less, you have to dissect it with micro pins and like uh, operating procedure for essential eyes and those kind of things are needed.
Yeah.
Precision is needed. Precision cuts are needed.
Right.
Everything's like that kind is needed. So you need very good microscopes and leisure time to
To do
do
that.
that.
But you need that means like an electron
Uh,
microscope or better quality
Electron
dissection
microscope is not needed for dissection. Dissection stereo microscope, which is
stereo
not
microscopes.
costly, but
Right.
you have to sit and give time.
Hmm.
That
Hmm.
is most important.
Right. So I think
And your mind should be at peace.
my suggestion would be that at least for this SCCS, you should put up a slug taxonomy workshop, uh, because there are many participants who come there where you might be able to find some masters or bachelor's students or PhD students who might be able to take this up on a more serious way, etc.
There are ample opportunities,
Yeah,
so
yeah.
they can.
Chinmay has asked another question. Have there been any visual differences in on the rocky shores between back then and now? How has the habitat changed, or is it the same in your opinion?
On
Chinmay,
my
which area?
opinion,
Can you tell which area specifically, or generally across? That's a very
no,
broad question, but yeah, Vishal, if you want to answer.
Like
generally,
Ratnagiri.
if we see Ratnagiri, then winters are shifting towards December. After December, ideally, that green algae and that growth should be there, but it's now shifting to first January or like that. So seasons are shifting. It's means maybe due to rain extension of rain into winters or that I don't know specific because now I am in winter. I am going to Lakshadweep, so I'm not here.
Right.
But based on whatever I gather
But
information
are there
from
changes in the winter in Lakshadweep as well in terms, do the local people?
no, their local people tells that so one that cyclone whenever comes, the wind shifts to other side means of island, I guess.
Mhm.
So during that shifts of winds, uh, their season changes and water stabilizes. but now that is also not happening. This year there was no cycle. Now that bleaching is starting.
Right.
So
So you'd say, matlab, are there sea slugs? You mentioned, of course, that they are found in sandy areas, but are they like obligately connected with coral reefs or uh,
no, no. What I said, we search more means since 1864 to 60s, I guess.
right,
Some pockets were studied in
deeper,
details.
deeper. Hmm.
Yeah, because those were coral reefs, so more attention
Hmm.
is given to those areas. Now, due to that, I naturalist and I like the citizen science things, more and more shores are being explored,
Hmm.
and that is good thing about citizen science, that
Hmm.
it's providing uh, equal opportunity liberally.
Right.
That's what I can say. But it also has that other responsibility. So single species may be observed by 10 individuals, and all 10 individuals are on iNaturalist, and they are showing same photo, I mean same individual's photo. It can be counted to number 10. That kind of means issues are there, but at least we are getting some pinpoints on maps as a distribution of records.
Right. So I'm going to combine Usha these two last questions because you've been at it for almost more than 1.5 hours. So
No
one
problem.
is that so she's mentioned Sea Slug Forum is Sea Slug Forum a reliable source to go check for taxonomic validity, or are there other sites? You said this before, but sorry if you could just repeat that once. Besides Sea Slug Forum,
Cis Luck Forum is a taxonomic validity. You can go to worms. There is one site, marinespecies.org. You can check older names there to get accurate at present whatever stabilized taxonomy.
hmm.
It is not accurate. Every time you have to go to
Keeps changing.
reliable research papers, and but whatever it's accepted by most of the editors, it is given there. So you can quote that reference and use that as a reliable taxonomic means input from there. There is other uh, index nudibranchia and bibliographia nudibranchia from by Gary MacDonald. He compiles list uh,
But
of
those are
all
largely
the
checklists, I think, right?
large, yeah, nudibranchs only,
Yeah,
not all other species, only
right.
nudibranchia group. So that is also reliable source. Sea Slug Forum is reliable because that
Visuals are there.
visuals, and he has answered it in very
Detail. Yeah.
detailed, and it's, no one has explained
I
sea
think he could
slugs.
put an amateur on at least a track of how to look at it in a more
Yeah, because
systematic manner.
I was not knowing anything
Right.
about sea slug
Yeah.
in
Yeah.
2009. I learned everything from there initially. Now everything is changed
Changed.
in terms of names,
Right.
but basics cannot change. How to identify, what to look for, how to look for, where to find them, everything is there.
And is there a which is the best time to go watch sea slugs?
During low tides, if you are going to rocky shores, on reefs, you can go uh, during low tide. If you are going to do intertidal walk, for snorkeling, low tide will be better because
Yeah.
column will be less.
Yeah.
So you can you don't need to dive deeper.
So two last questions from my side. One is that are there any sea slugs that people should be careful of? That not to touch, not to get in contact with, or are they like potentially highly venomous, or that they're
Uh, there are some species which secrete acids, like Dendrodoris tuberculosa, but those are not dangerous. Means like
extreme?
Conus and other,
Right.
not venomous. You will not die
Right.
directly. You will get some time in between to apply some. solutions,
Right.
rub it off or rinse it with water. If
Hmm.
you eat it
Yes.
directly,
Yeah.
if you are insane enough to eat it, then
Yeah. God bless you. Right.
uh, so
Second and last question is that what do the fishing community or the local people have if they see you, are they aware of sea slugs, or do they know about it, or if they see you looking at it or whatever, do they have names for it, do they have any identity, like what is the there, where do they stand as far as the local communities, fishing communities are concerned?
local communities know where to find some species like sea hares and
Right.
whichever is large.
Large,
If
right.
you ask them like where we can find this,
Ah,
they can tell this is found in this particular area during this season. But mostly large species. For means some dive institutes also can suggest some areas like you will find this colorful species because they are doing macro photography and some people are coming only for photographing sea slugs.
right.
So
And any final advice to the audience as to how, what, from a citizen science perspective, how would you want them to collect the data? What do you think will help knowing more? As you said, there are 100 more species probably to be described, and so how do they go in data collection so that that can be done?
collect all possible data means at least photograph all possible angles. On iNaturalist, we upload only one or two photographs, but if you upload from all possible angles, it will be easy for any expert.
Hmm.
I'm not saying I'm expert, but there are many experts better than me, and they can identify it carefully and clearly.
Also, nowadays there are these macro magnifying sort of attachments you get with even camera, mobiles, and other things that you can use to zoom in
Oh,
into the.
now cameras also, mobile cameras also very
Yeah,
efficient.
yeah,
So if you zoom in with that camera, use 45-degree angle so that reflection of any other surfaces won't come.
right.
Use some torches. You can take photo even from outside water to avoid
Any,
that
any
reflection effect.
bioluminescent sea slugs? Any bioluminescence found
Uh,
in them?
that on Sea Slug forum there is one post on this, but that from our region, Plocamopherus silonicus, if it's means threatened to life, then it emits
Oh, I see.
light from extra branchial appendages. So I haven't seen it because I haven't threatened it for life yet. So, but
Okay.
there are records
Okay.
mentioned in that.
Yeah, I think there is a lot more. I think maybe another year or two later, we'll have another session with you to understand more what all more has been. But I would tell the audience that I think, it's now their responsibility to collect systematic information and collate it well, share it, with the respective experts that are there with Vishal and others. And,
You can
build his organization, build his team bigger so that we have more researchers who are collecting this data and organizing it in a systematic manner. Yeah, you were saying, Vishal?
You can share my what that
Email address, phone
email
number?
address or phone number, whatever
Yeah.
means
Okay.
you can. So if they want to know more, they can
Could,
connect.
could you dictate? I will type it out.
Should I add it in chat?
Yes, please, if you can.
No, it won't be public. I don't know. It's recording, right?
Yeah, it is recording. Yeah.
YouTube. That's why I'm asking.
Uh, it's up to you. If you want, we can then send it. All the registered participants only, we can send it to them via email. If you don't need to share it here, then.
Okay,
Yeah.
you can.
Okay.
That will be much
I'll do
better
that.
because spam and everything will.
But that having said, I request all the participants, please be patient with all these, speakers, because they are out in the field sometimes at 1 month, 2 months at a time. Don't get impatient and say, I wrote to you 1 week ago and you've not replied. They have a lot of work to do, and they sometimes can be in field for 2 to 3 months. So please be patient and please be respectful for the support that they're giving you, because it's a voluntary activity that they're doing. Don't act, privileged that, oh, I have to, because I've written to you now, I need to get a reply. And ask your questions, in a more systematic manner. Try to train yourself to ask systematic questions, some of which you have done today. So thank you, Vishal. I would ask Rahul to formally thank you, but yeah, thank you very much. I think everyone will agree this is one of the most engaging sessions we've had. Some wow images. I'm sure many of these people will go back to our YouTube channel, the Green channel, and see this video over there. We'll be editing it and putting it up over there. So yeah, please visit the channel and follow us and like us. And thank you once again, Vishal, for accepting our offer to.
Thank you
talk.
for
What do you know?
opportunity.
Yeah.
Um, Once again, I would like to formally thank you for giving us your time and sharing your experience, insights, and so many lovely photos with us. Sea slugs are something that not a lot of us know about. Also, not something which are very well known among the people. So it was really great that we could come on this journey with you and learn so much about these interesting creatures. Similarly, I would also like to thank the audience for tuning in and being very participative and asking all the right kind of questions. And it is this collaboration with both the speaker and audience that has made these talks so successful. And we hope for continued participation in the future. So thank you, everyone.
And I'd like to announce that on the 11th of May, we'll have our next session of the Sprouts lecture series. I have a sort of a vague confirmation for the next speaker, but I don't want to announce it at this point. So, but please look out for our 11th May, lecture, and please join us again. Thank you.
Thank you all.
So have a good weekend, and we hope to see you. Also, I would like to announce about SCCS that SCCS is now started to accept papers and presentations for the conference, Students Conference for Conservation Science, which will be in October from 21st to 25th. So those of you all who are doing any research in any taxa or field, please send in your papers, please send in your poster presentations and all for SCCS, and hope to see you in Bangalore also at that point of time. Yeah, great. Thank you, everyone.